discuss antigens. In 1899, Dattray named antigen as a substance between bacteria and antibodies, also called as immunogens. He believed that antigens are precursors of antibodies. Similarly, zymosin is a precursor of zymase. Zymase is a mixture of enzymes. He also believed that antigen induce the production of antibodies. Antigen is a substance that evoke the immune response to produce antibodies and react with specific antibodies. Antigen is a molecule such as may be present at the outside of the pathogen. The AG abbreviation stands for antibody generator. In the diagram you can see how antigen induce the immune response by interacting with an antibody that matches the antigen's molecular site. Classification of antigens. Based on immunogenicity. That means based on the power to provoke an immune response, that is immunogenicity, which can induce antibody formation and can react with these antibodies, called as complete antigen. Incomplete antigen, also known as haptenes, unable to induce antibody formation on its own, but when linked with a carrier protein become immunogenic. Subdivided into two types, complex and simple. On the base of origin, antigens are divided into exogenous, endogenous. Exogenous antibodies enter the body from the outside like inhalation, ingestion. Endogenous antigens generate within previously normal cells as a result of normal cell metabolism or because of any kind of infection. Autoantigens. Autoantigens is a normal protein or complex of proteins recognized by immune system of a patient suffering from autoimmune disorder. Under any normal condition, these antigens should not be the target of the immune system. Immunological tolerance is the failure to mount an immune response to an antigen, and if immune system should respond to self, an autoimmune disease may result. Isoantigen that stimulate antibody production in different members of the same species. Heterophile antigen are antigen of similar nature and shared by different species. Factors of antigenicity. Foreignness. Antigen must be foreign to the individual to induce immune response. According to Burnett's, non-self means substance that never contact with lymphocyte during embryo period. size. Larger molecules are highly antigenic, and antigens with lower molecular weight are weakly antigenic. Chemical nature. Most antigens are protein or polysaccharides, but proteins are more effective antigens. Susceptibility to tissue enzymes. Substances which can be metabolized and are susceptible to the action of tissue enzymes behave as antigens. Substances which cannot be metabolized in body are not antigenic. Antigenic specificity. It depends on epitope. Epitopes are antigenic determinants, smallest unit of antigenicity. Each epitope induces a specific antibody formation. Species specific. Tissues of all individuals in a species possess species specific antigens. It depends on isoantigen. A species may be grouped on the basis of presence of isoantigens in its members, for example, blood grouping. That depends on erythrocytic antigen. Self-antigen are non-antigenic. These are lens protein and sperms. These are not recognized because they develop after embryo life as sperms. Organ specificity. These are confined to a particular organ. Some organs to different species share the same antigen, as brain specific antigen are shared by man and sheep. The same or closely related antigen present in different tissues of more than a species are known as heterophile antigen. For example, Forceman antigen. It's a lipoprotein polysaccharide complex widely present in man, animal, birds and different bacteria. Superantigens. Superantigens are a class of antigen that result in excessive activation of the immune system. Only a small fraction of the T cell population is able to recognize the antigen and become activated. Some antigen which polyclonally activate 25% of T cell are called as superantigen. Different tests for antigen detection used in diagnosis. Direct ELISA, direct immunofluorescence, RIA, radioimmunoassay, neutralization test, CFT, complement fixation test, and immunohistochemistry. It's a immunostaining technique. Thank you.